The overall goal of this procedure is to observe the dynamic changes in the protein FtsZ during cell division in bacteria. This is accomplished by applying Bacillus subtilis cells expressing the FtsZ GFP fusion to the surface of a small agarose pad. The pad is then inverted and transferred to a petri dish with a glass cover slip bottom. The second step is to acquire time-lapse images of the bacteria during cell division using a 3D structured illumination microscopy, or 3D-SIM, imaging system. Next, the quality of the raw data is assessed, and images with greater than 30% decrease in intensity due to photobleaching are discarded. The final step is to quantitatively analyze the changes in the protein localization during cell division by measuring the fluorescence intensity distribution of the FtsZ GFP protein over time. Ultimately, fast 3D SIM is used to show how dynamic the divisome proteins are during cell division in bacteria.
The main advantage of this technique over existing techniques like confocal microscopy is that we get three-dimensional information about the changes that occur in cell division in live cells.
Though this method provides insight into bacterial cytokinesis, it could also be applied to other systems where we want to look at dynamic protein changes, such as in mammalian cells.
We first had the idea for this when we saw traditional 3D SIM images of fixed bacterial cells showed that the FtsZ protein was not homogeneously distributed around the Z ring. We wanted to further examine if this distribution was linked to changes that occur during Z ring constriction.
Generally, individuals who are new to this technique will struggle because of a failure to match the refractive index of their oil to their sample, and this will lead to